In August of 2026, a genetics paper landed in Scientific Reports, one of Nature's own journals, and the mainstream barely noticed. The same researcher who first pulled DNA from the Shroud of Turin just went back to the same samples. Different method, sharper tools, the kind that doesn't copy DNA before reading it. It reads whatever is sitting in those threads raw, exactly as it is. The ancient Middle Eastern signal didn't fade. It got louder. New organisms showed up, microbes from the Dead Sea, coral from the Mediterranean, cultivated plants from the ancient world. A biological trail that runs straight through the geography the Shroud's history has always claimed, and that a medieval forger in France had no access to. Nobody published a rebuttal. Nobody offered an alternative explanation. Just quiet. Sound familiar? Because this is exactly what happened the last time a lab aimed better technology at this cloth. And the time before that. And the time before that. The Shroud of Turin isn't ancient history. It's an open forensic file. And it just got reopened. Again. Here's what happened, and why nobody's talking about it loudly enough. Gianni Barcaccia, geneticist, University of Padua. He's the same man who ran the first DNA extraction from the Shroud years ago. The study that found genetic markers from ancient populations across the Middle East, North Africa, and South Asia woven into the cloth's fibers. Skeptics pushed back hard on that one. Fair enough. The method he'd used, PCR amplification, copies tiny DNA fragments millions of times before reading them. That copying process can also multiply contamination. So the objection was simple. Maybe the ancient signal was just noise, boosted by the machine. Barcaccia heard that objection, and he came back with a method that removes it from the conversation entirely. PCR-free next-generation metagenomic sequencing. No copying, no amplification, no multiplication of anything. The machine reads whatever DNA is physically present in those fibers, exactly as it sits, without touching it first. Think of the difference like this. The first test was sharpening a blurry photo on a computer screen. This new test was building a better camera. He went back to the exact same samples, the ones collected during the official 1978 STURP examination, stored under controlled conditions for nearly 50 years. And the better camera found more, not less. Multiple human mitochondrial lineages. Still there. Still not one population, a mix of ancient lineages from across the Near East. But this time, the sequencing picked up things the older method had missed completely. Halophilic archaea, microorganisms that only survive in extremely salty environments, the kind found near the Dead Sea. DNA from Mediterranean red coral, a species found almost exclusively in the coastal waters between southern Europe and the ancient Near East. DNA from cultivated wheat, domesticated cattle, chickens, dogs, cats, biological traces consistent with a cloth that traveled through agricultural regions of the ancient world. Not a workshop in medieval France. The researchers said it directly. None of this proves the Shroud is 2,000 years old. But here's what they didn't say, and what the data screams if you're paying attention. The contamination argument, the one skeptics had relied on for years, just lost its foundation. Because PCR-free sequencing doesn't amplify noise. If anything, it should have returned a weaker signal. Instead, it confirmed the original findings and added layers that nobody had detected before. 2 studies, 2 methods, same samples, same direction, years apart. Hold that. Because to understand why this matters, you need to see what was already in the file before this paper ever landed. A sealed chamber inside Turin's cathedral. The smell of old stone and cold air. 40 scientists, physicists, chemists, imaging specialists, 1 photographer who'd spent years working on projects alongside NASA, standing around a 14-foot strip of yellowed linen with 1 assignment: find the medium. Whatever a forger had used to paint this image, oil, tempera, iron oxide, charcoal, identify it, document it, and close the book. The photographer was Barry Schwartz, Jewish, skeptic down to his bones. He told friends before the trip it would be quick work. Expose the hoax, see some of Italy, fly home. He stayed for 5 days. He spent the rest of his life on it. When the cloth came out under laboratory lighting, the room went still. Schwartz started shooting at extreme close range, hunting for the microscopic ridges and pigment traces that every known painting technique leaves behind. Frame after frame, fiber after fiber, nothing, not in visible light, not in ultraviolet, not in infrared. 40 scientists, 120 hours, and not one of them found a forger's fingerprint anywhere on the cloth. Then, somebody fed the image into NASA's VP-8 image analyzer, hardware built to convert flat satellite photographs into three-dimensional terrain maps of other planets. Normal photographs produce garbage in a VP-8 because regular images don't carry real depth information. A painted shadow is an artistic choice, not a measurement of distance. The Shroud's image produced a clean, anatomically correct three-dimensional body. Every brightness level on the cloth corresponded to a precise physical distance between the linen and whatever had been underneath it. The cloth had captured a body's shape the way a flatbed scanner captures a document pressed against glass. Under the microscope, it got worse. The image sits only on the absolute outermost surface of each fiber, a layer thinner than a single blood cell. Touch it with a blade and it's gone. Clean white linen underneath, nothing absorbed, nothing spread. No dye, pigment, or ink from any era in human history produces a mark that impossibly thin. The only explanation that survived review, a sudden intense flash of radiant energy, powerful enough to change the chemistry of the linen surface, but so brief it never burned through. No laboratory has reproduced it, not once in nearly 50 years. Schwartz later said the silence in that room was suffocating, like the cloth was daring them to explain it, and that was just the image. The blood was about to make everything harder. For 2 decades after that investigation, the blood was the skeptic's last card, and it was a good one. Red stains, bright, vivid, almost wet-looking. And anyone who's taken a first-year forensics class knows that's wrong. Real blood darkens within hours, ancient blood crumbles to black powder, red blood on a cloth that old is forgery. Some amateur with a pot of cinnabar paint faking wound marks for a medieval crowd. Good theory. Clean logic. One man destroyed it with a single finding. Alan Adler. Blood chemist. Not a believer, not a pilgrim. A scientist who studied how blood breaks down at the molecular level, period. He analyzed the Shroud stains expecting to find iron oxide pigment and closed the file. Instead, he found something that made him pick up the phone in the middle of the night and call Schwartz. Bilirubin. Abnormal concentrations of it, far beyond anything a healthy body produces. Here's what that means, and this is the hinge of the entire cold case. When a human body undergoes hours of escalating catastrophic physical trauma, the liver begins shutting down. As it fails, it dumps bilirubin into the bloodstream where it bonds chemically with hemoglobin. That bond is permanent. It locks the blood red. 100 years, 1,000 years, 2,000 years, it will never darken. The red on this cloth wasn't a mistake. It was a molecular record of how someone died, slowly, over hours, with organs collapsing before the heart stopped. Adler also confirmed human hemoglobin, human albumin, and human immunoglobulin in the stains. Other researchers mapped over 100 wound marks across the back of the figure, consistent with a multi-tailed Roman whip built to strip skin from muscle. Puncture wounds through both wrists, a wound in the side matching the profile of a Roman military lance. Soil, traces near the feet carrying a mineral signature specific to the limestone bedrock underneath ancient Jerusalem. And underneath all of it, one detail that still has no explanation. The blood reached the cloth first, the image formed second. Every spot where blood had soaked into the fibers, the image is absent. Whatever energy event burned that three-dimensional imprint onto the linen, it stopped at the exact edge of every bloodstain and went no further. As if it knew where the blood was, as if it went around it. Which piece of this evidence do you think cracks the case? Hold that answer, because now you need to see what the DNA looked like before August of 2026. When sequencing technology first got sharp enough to pull genetic material from the Shroud's fibers, researchers expected to find a mess. Hundreds of years of bare hands touching this cloth, priests, conservators, pilgrims, restorers, should have left a genetic pileup of European DNA drowning out anything older. The results came back, and nobody said a word. The genetic material recovered from inside the fiber structure, not the surface, not the handling layer, but embedded within the threads, carried markers from populations across the ancient Near East, South Asia, and North Africa. Some of those lineages haven't existed as living populations for thousands of years. A cloth manufactured in medieval Europe should carry a genetic profile screaming Western European. This one doesn't. The fingerprint reads like a census of the ancient world, and the damage pattern in the bloodstain DNA was unlike anything natural aging produces. Instead of the slow, even breakdown you'd expect over centuries, a book fading page by page, the chromosomes appeared to have come apart from the inside all at once, as if every cell had been disrupted by the same event at the same moment. Geneticists associate that pattern with one thing: catastrophic biological trauma at the cellular level. That was the first study. Fair objections were raised about the method, and now, years later, the same researcher has answered them, with a tool that doesn't amplify, doesn't copy, doesn't boost. And the signal held. The Dead Sea microbes are new, the coral is new, the agricultural plant DNA is new. Every layer, the sharper instrument found points in the same direction the original data pointed, just with more detail and less room to argue. Every year, the instruments get sharper, the sequencing goes deeper, the imaging gets more precise, and every time a better tool's aimed at this cloth, the picture doesn't simplify. It gains another layer, as if it was built to reveal itself only as fast as we're capable of understanding it. We can photograph black holes, we can land machines on other planets, but nobody has successfully copied this cloth. Nobody has explained the image. Nobody has accounted for blood chemistry that shouldn't exist. And nobody can explain DNA from a vanished world, confirmed now by two independent methods, that was shattered from the inside by an energy event modern physics doesn't have a name for. Barry Schwartz, the skeptic who flew to Italy expecting to debunk a relic, spent decades after that trip cataloging every piece of evidence the investigation produced. Never took a cent for it. He said once that the data led him somewhere he never planned to go. If the bilirubin records hours of catastrophic torture, if the DNA confirms ancient Middle Eastern origin, now twice with two different methods, if the chromosomes were torn apart from the inside by something nobody can identify, what left a three-dimensional image on burial linen without burning it, in the same instant it destroyed human DNA at the cellular level? The Shroud of Turin isn't a closed case. As of August 2026, it's less closed than it's ever been. This is Hidden Latitude. We go back for the cases everyone else assumed were already finished.